So it's very nice to come and talk to you today about some work that we've been doing over the past 3 or 4 years, which is about how bacteria aggregate into clumps and also how they make biofilms. So actually the work that I'm going to present has been the efforts of international collaboration, and I'll mention at the end we've been doing the computer simulation part of the work, but there's also been experimental work which has been done by our collaborators in Copenhagen, in Austin, Texas, and also in Nottingham in the UK, and I'll mention them all at the end. But I wanted to highlight the postdoc from Edinburgh who's done almost all of the work that I'm going to present mainly, which is Gavin Mellock. So this is mainly his work. So what are these bacterial biofilms? So biofilms are communities of bacteria that grow on surfaces, and here's a picture of one. This is a picture of a biofilm growing on a medical implant. And why are they important? Well, one reason they're important is because it's believed that most of the bacteria in the natural environment, actually the way that they live, is as these surface-attached communities, even though in the lab people have historically studied bacteria growing in well-mixed liquid cultures. Actually, it's believed that's not really how they live in the natural environment. But also from a human perspective, biofilms are important because they can cause chronic infections. So, for example, when a biofilm forms on a medical implant, it can be very difficult to remove it using antibiotics for reasons that are not actually very well understood. And also from an industrial perspective, these surface-attached bacterial communities can cause problems like blocking pipes or also damaging the surfaces of industrial devices. So they're important economically and also medically. But from a sort of physical science perspective or mathematical science, we like to study biofilms also for their fundamental beauty, and we feel that these biofilms. So this is a picture of a biofilm which has been taken in a confocal microscope, and it's been grown in a flow cell. Here you see it's a little plastic device where you can flow nutrient medium through the device while you actually image it in the microscope, and the biofilm grows on the bottom surface, and you can actually track in the microscope in 3D how the spatial structure of this community forms, and you can see that the images that you get are really quite beautiful. And so of course this is a non-equilibrium system because the bacteria are growing, and that's fundamentally a non-equilibrium process. And so this is an example of non-equilibrium self-assembly which we can actually track in time and space in the lab. So that's also a beautiful thing. So what's going on when these biofilms form? What processes are controlling the structure that you get? And so the major process which we think is important and also which we focus on in our research is that there's an interplay between the bacterial growth and local gradients of nutrient concentration. So typically the nutrient in these experiments is diffusing. For example, in this experiment, the nutrient is supplied in the liquid medium that flows across the biofilm, and so it reaches the bacteria by some kind of flow transport diffusion process. And then it gets consumed by the individual bacteria. And so because of that, there are spatial gradients of nutrient that form. And bacterial growth is coupled to those nutrient gradients because the bacteria grow faster where the nutrient's more abundant. And so there's an interplay between the spatial structure of the nutrient concentration and the spatial structure of the bacterial population. And that's important. You can see it's important from this example, which is taken from some work by Kevin Foster's group in Oxford, where they just have done simulations of a biofilm growing at 2 different nutrient concentrations. You can ignore the red and blue, that doesn't matter. And the only thing to take from this is that low nutrient concentration, you get a completely different structure predicted from the simulations for the biofilm compared to at high nutrient concentration. So it's clear that this interplay between the spatial structure of the nutrient and the spatial structure of the population is important. So that's the main focus of this talk, but also it's important to realize that this is a biological system, and so it's complicated. And there are many, many factors which actually we mostly don't take into account in our models, but towards the end of the talk I try to explain how we're trying to take them into account in our current work. One of them is the fact that these bacteria actually, many of them are producing sticky polymers, and so the role of these polymers is not completely known, but it's believed that they function in sticking the bacteria to the surface and also to each other. But also they might form a kind of protective coating around the biofilm, and that might be the reason why antibiotics are not very effective against these biofilms. It's also known that bacteria in these biofilms typically do a lot of signaling, which controls all kinds of behaviors. They can be motile, so For example, you might get bacteria that actually move around on the surface of the biofilm and take themselves to the top of the towers. And also there's believed to be, in some cases, there might be kind of programmed cell death in these biofilms. So it's a complicated system. I think it's relevant for this course about multiscale integration, because clearly what you have in this system at the molecular scale, you have to understand the transport and diffusion of the nutrients and also possibly signaling molecules. So on the molecular scale, you have molecular diffusion and transport, but then that feeds into the scale of bacterial cells, because you have bacterial growth and behavior, which depends on the nutrients and the signal molecules. And then all of that feeds into the creation of these larger scale multicellular spatial structures. So it's a really nice example of multiscale integration in a biological system. So in Edinburgh, as part of this international collaboration, we're doing the simulations, computer simulations. And so how do we actually make models to describe the formation of this spatial structure? Well, there are different ways to do it. So one way, which we actually haven't done, is to construct continuum models. So in those models, you basically construct a grid in your simulation, and you set up a set of partial differential equations which describe the nutrient concentration fields, and also so that's one partial differential equation, but then there's another differential equation which describes the actual biofilm, so the density of mass of bacteria. And if you do that, then you can do numerical solutions and you can get some really interesting results. So this, for example, is an image taken from a paper which describes the formation of streamers in a flow field. So in this simulation, there's a nutrient which is flowing in the right-hand direction, and the biofilm is this white thing which is forming on the surface of these beads. And you can see that there's a coupling between the sort of wobbling motion of the biofilm in the flow field and the nutrient concentration field. So the advantage of these continuum models is that you can easily include effects like fluid flow, which you simply include in your partial differential equations. And actually, we're thinking of doing that in some future research, but at the moment, we've focused on the other way of simulating biofilms, which is individual-based models. And those create images a bit like this. So in those models, rather than representing the bacteria as a continuum field, you actually represent individual bacteria as particles. And so in this simulation here, which is from Cristian Picurianu's work, individual bacteria are represented as little spheres. In this particular case, they can take two different types, the yellow ones and the blue ones. And what happens is that the individual bacteria, they can grow and divide and form spatial structure. And their behavior is coupled to a grid which has a local nutrient concentration on it, which is again represented as a partial differential equation. So these are two different ways of modeling bacterial biofilms. What we've been doing in our research is the second way, as I described. So we have these individual-based models, and we've actually been using a software package which is designed to do this, and it's called iDynamics. We've been using this package, and what it does, as I said, is individual bacteria are represented as spherical particles, and the food is represented as this continuum concentration field. And so an individual bacteria, what it does, it consumes food, depending on its local concentration. It grows, so that means the sphere expands. And when it gets to a critical radius, it divides in two and makes two bacteria. And also, individual bacteria are designed so that they can't overlap. So there's a steric interaction which causes the whole biofilm to expand. And the nutrient diffuses and is consumed by the bacteria. So in a bit more detail, what happens? So the bacteria, this is about how the bacteria grow. So they consume food, and actually the way that they consume food is described by this equation. So here X is the mass of a bacterium. S is the local concentration field, and what you see here is that this equation is actually nonlinear. So when the concentration of the food is low, then there's a linear relationship. So this thing becomes more or less linear between the growth rate and the nutrient. But when you get too much local nutrient concentration, then the function saturates, which means that there's a limit to how fast a bacterium can actually grow. Okay. And then once the bacteria reach critical concentration, as I said, they divide in two. And also you have these mechanical interactions. So at each time step in the simulation, you do this growth reaction, and then you check to see whether you generated any overlaps between the bacteria. And if you detect some overlaps, then you randomly shuffle the cells about in a shuffling algorithm to try to remove the overlaps. So this is supposed to be a mimic for steric interactions between the bacteria. Of course, it's not a very good mimic because it doesn't include any forces. It's just a random shuffling thing. And one of the things that we're hoping to do in future research is to actually improve on this mechanical interaction description. And then the nutrient, as I said, you have a concentration field for the nutrient, and it diffuses from above. It gets consumed by the bacteria. And we actually assume, or iDynomics actually assumes, that the diffusion of nutrient is rapid compared to the rate at which bacteria grow. And so in each time step, what you actually do is solve the steady state reaction diffusion equation for the nutrient. And that's quite an efficient thing to do. And usually, we believe that it's pretty OK to do that. So what I just described is what we've been doing in most of the research that I'm going to present. But I think it's good, since this is a sort of a tutorial type event, I wanted to be honest and explain that this is not actually perfect. So there are lots of ways in which we actually don't like the way this software operates. And one of the ways is that, as I described, is that it has, in order to mimic steric or physical interactions between the bacteria, it does this shuffling algorithm. And that's kind of unsatisfactory because, as I just explained, many bacteria are actually sticky. And so you have these physical forces between bacteria which are not necessarily repulsive. They could also be attractive. And so that's something which we need to find ways to implement in a different way from what we do currently. Another thing is that in iDynomics, one assumes that the bacteria are spherical, at least in 3D, whereas in reality, in many bacteria are not spherical. So, for example, Pseudomonas aeruginosa, which is a model system for biofilm formation, is actually rod-shaped, and we don't know how much effect that has on the spatial structure formation. Also, this method doesn't include motility. It assumes steady-state nutrient concentration, and perhaps also importantly, it doesn't include fluid flow. So in the microscope setup that I described earlier, there's actually a fluid flow across the surface of the biofilm. In the microscope, that's usually slow, and so it doesn't probably matter very much. But in many real applications, so for example, if you imagine biofilms forming on a heart valve inside on someone's heart implant, then there's going to be quite a rapid flow of blood across the biofilm, and so it actually can be important to properly include fluid flow. And also the code runs slowly. And for that reason, it's kind of common to run the code in 2D instead of 3D, which is somewhat unsatisfactory. So having said all this, we still are using this software package, and the reason is because the one thing that it does do correctly, we think, is that it couples bacterial growth to a spatially varying nutrient concentration field. And so what we're doing in this research is asking the question, what are the consequences of a coupling between a growth of a bacterial population and a local nutrient concentration field? So using this one very simple physical mechanism, how much can you explain about what happens in a bacterial biofilm? Okay. I should mention that my colleague in Edinburgh, Bartek Václav, actually is developing a different software package of his own, which takes a more physical approach. So he actually does include rod-shaped bacteria, and he also includes more physical description of the interactions between the bacteria and also frictional interactions either between bacteria or between bacteria and the surface. So hopefully that's something that we're going to do better in the future. Okay, having explained all that, what I plan to do in this talk is to present three different topics that we've been working on. So the first topic, which is going to take up most of the talk, is some work that we've actually more or less finished, which is about how the spatial structure of a biofilm depends on its initial configuration. And then afterwards, hopefully if I've got time, I'm going to present very quickly two different topics that we're just starting to work on. So those ones are really preliminary work. So for the first topic, what we've been thinking about is the role of the initial configuration in the formation of a biofilm. So I guess hopefully you know that when you have an equilibrium system, so for example a system of colloidal particles at equilibrium in a temperature heat bath or something like that, it actually doesn't matter how you set up the system at the beginning of your experiment, because if you wait long enough, you expect that the system will on average reach the same final configuration. And that's called the ergodic hypothesis. And what it says is if you wait long enough, as long as the system samples all of phase space, then it will just reach the same, on average, final configuration. But as I said, a biofilm is not an equilibrium system, and bacteria are growing, and that pushes them out of equilibrium. So our question is, what happens for biofilm? So if you initiate biofilm growth with different initial configurations, does that have a long-term effect on what you observe at the end of the experiment? So why should we care from a biological perspective? Well, it turns out that it does sort of matter. So the classic picture of how biologists think that biofilms form is illustrated here. And what's supposed to happen is that you have these individual bacteria swimming about in liquid medium of some kind, and every now and then one of them attaches to the surface. And once it's attached to the surface, it starts growing and dividing and making little microcolonies, and also producing sticky polymer that sticks it to the surface. And then you get these kind of structures developing, which consist of bacteria plus sticky polymer. And then at some point, after they get big enough, there's a dispersal event, which actually is not particularly well understood. And the biofilm sheds off these new single cells, which then swim off and go and form new biofilms somewhere else. Okay. So the classic picture of biofilm development assumes that there are these individual cells, which are a kind of stage in the developmental process. Okay. But what's also observed is that many bacteria, if you just look at a liquid suspension of them, it contains many preformed aggregates. So here's a picture of a preformed aggregate that formed in a liquid suspension. And these aggregates can actually be quite big. So they can be tens of thousands of cells. And they're kind of ubiquitous in liquid suspension. And so the thinking is that these aggregates actually might sometimes actually seed biofilm growth. So it could be that the biofilm doesn't grow from individual single cells, but it actually grows from a mixture of individual single cells and preformed aggregates. And so, so our question was, if you take account of these aggregates in the seeding of the biofilm, how does the picture of biofilm development change? And so to link that back with my question about the initial configuration, the question is, if the initial configuration isn't actually individual dispersed cells, but it's a mixture of dispersed cells and an aggregate, what difference does that make? Okay, so I mentioned that this is a collaboration, and so these are some results from our collaborators in Copenhagen who actually do these flow cell biofilm imaging experiments using Pseudomonas aeruginosa. And what they discovered in these experiments is that the presence of an aggregate at the beginning can make a big difference in some conditions. So these are some results of an experiment where the initial configuration, which you see here, was of individual bacterial cells. So there's no aggregates in this initial setup. And then this is a picture of the same part of the flow cell 24 hours later. And what you see is it's formed a kind of biofilm with some kind of rough spatial structure. But this is another experiment. This time it started with an aggregate in the middle of the field of view, surrounded by these individual cells. And what you can see is that after 24 hours, which represents many, many, many generations of these bacteria, the aggregate is actually very large. And so the bacteria which initiated in this aggregate have actually proliferated and come to be important in the spatial structure of the biofilm. So what the experiment suggests is that at least under some conditions, these aggregates can have an important effect, yeah?
So is this the steady state of the biofilm?
No, it doesn't. It doesn't have a state. Well, it's still growing. Yeah, so you have to pick a time which you say you're going to image at. Yeah. Whether or not it has a steady state is an interesting question. So, but in this case, no, it's still growing.
Sorry?
Yep.
Do you grow faster if they are fixed to the aggregate in the middle of the chamber?
I'm going to discuss that. So sometimes yes and sometimes no is the answer.
So if you really do patterning of the micro chamber, like with spikes in the middle of the chamber, something like that?
What, you mean have the surface be bumpy?
Yes.
Yeah, we haven't done that, but it's a very good question. Yeah?
So in the situation where you start with no aggregate, if you kind of consider every individual cell a mini aggregate,
Yeah.
you get correlations with the bumps in the
Oh.
simulating the.
That is a good question. I don't know the answer to that. It's a good question. I think maybe we should check. Yeah, but I don't know the answer. Any more questions? Okay. Okay, so what we actually did in Edinburgh with Gavin, our postdoc, is simulations. And so he tried to reproduce this experimental scenario using computer simulation. So actually, I don't know if you could see very well, but the way the initial state of the simulation was lots of little individual cells here, which are actually shown in red, dispersed across the surface. And then in the middle of the simulation, he created an artificial aggregate of identical cells, but we label them in green to show that they initiate in the aggregate. And it's assumed to be round. And then he just runs the simulation for a length of time and sees what the final configuration is. And here's a representative example from one of his simulations, where you can see that actually in this simulation, the progeny of the aggregate have come to be important in the spatial structure of the biofilm, because the aggregate has many descendants, and it's actually, on average, they've done better than the surrounding single cells. And you can also see from this image that the nutrient concentration field must have played a crucial role in these simulations, because you have this area around the aggregate where the nutrient is depleted. And so there's been a competition for nutrients going on between the aggregate and the surrounding single cells. So it seems what we learned from this is that at least these simulations can, to some extent, reproduce what we see in the experiments. But of course, with simulations, that you can run them a lot more easily than experiments, much quicker and in more replicas and so on. And so what Gavin actually did was a whole series of simulations where he varied the density of these surrounding single cells. So from a regime where what we called low competition, so that means that there's only a few of these individual surrounding single cells, so that's on the left side of this graph, to a regime that we called high competition, which is where you have many of the surrounding single cells, and that's on the right-hand side. And he ran many of these simulations, and what he tried to quantify in the simulation is how well does the aggregate do compared to the single cells. So the thing that he's plotted here is the ratio of the number of progeny coming from the aggregate divided by the number of progeny per cell coming from the single cells. So if this number is bigger than 1, then it means that the aggregate somehow won. So on average, the cells in the aggregate had more progeny than the single cells. And if the number is less than 1, then it means that the aggregate actually was outcompeted by the single cells. And so, and these data are for different time points during the simulation. But what you can see from this data, broadly speaking, is that the higher the density of the surrounding single cells is, the more likely it is that the aggregate is going to win. So these curves increase. And also, if you wait a long time, then the aggregate tends to win. So what we learn here is that the outcome of this simulation is dependent on the level of competition. If there's high competition, the aggregate does better relatively, and if there's low competition, then the aggregate tends to do worse relatively. Okay.
Does it also depend on the density in the aggregate?
We didn't check that.
So, but you try to model, like, do the same as the surrounding cells density?
No, because the surrounding cells are sort of a 1D density, whereas the aggregate is a 2D density. No, what we did is we tried to make it a kind of realistic value. So you know from the experiments roughly how dense the cells are in an aggregate, and we basically tried to match that in the simulations. But it's true that we didn't try varying it, so that's an interesting question. Yeah?
You know the time scales in the last graph?
Yep, they're very long, I know.
How are they in relation to actual growth?
Well, so the experiments that I showed in the first, in the experimental slide were 24 hours. So that's kind of in between the red and the black. The reason that Gavin did these really long simulations is because he's interested in the kind of long time behavior, and so he said, let's just run it a really long time and see what happens. And you can run a flow cell experiment for a very long time as well, although he didn't. so yeah, there's a bit of a mismatch, but that was because he was really interested in seeing what happens at very long times. Okay, so from the simulations, you can actually analyze not just the configuration of the bacteria, but things like the flow, things like the concentration field of the nutrients, and also you can plot maps like this. So what this actually is a map of the growth rate of the individual bacteria in the initial configuration. And so that gives you some clues about what's actually going on in these simulations. So what we discovered from doing that is that in the high competition regime, so when you have many of these surrounding red cells, there's kind of two factors that contribute to the outcome. So one is that the individual single cells have a harder time growing because they have to compete with each other for the nutrients. So the average number of progeny coming from a single cell is anyway less if they're at high competition because they're all having to compete. But also the cells at the top of the aggregate have an advantage because they're closer to the nutrient source. And so when you plot this growth rate distribution, you see that the growth rate is the highest for the cells that are right at the top of the aggregate. So we think that the reason the aggregate does well under conditions of high competition is that firstly, the single cells do worse, and secondly, the cells that are at the top of the aggregate have this nutrient advantage because they're higher up. So that's why the aggregate comes to dominate, we think, yeah?
So do you also, so have you tried also reproducing these results by varying the nutrient concentration?
Yes. So yes, you can. We have varied the nutrient concentration, and basically the qualitative results don't change. But if the nutrient concentration's lower, then you get more pronounced effects. So obviously, yes. So it does what you would expect it to do. In the low competition regime, so what we see is that the individual single cells actually do better than they do in the high competition regime because they don't have to compete so much for nutrients. But also another factor comes into play, which is that in this aggregate, many of the cells in the aggregate actually never proliferate because they're stuck in the middle, and the cells in the middle of the aggregate have poor access to nutrients. And so we think that there are two reasons why the aggregate does poorly in the low competition regime. One is that simply that the single cells do better, and the other is that if you're in an aggregate, you have a sort of a disadvantage compared to the single cells, which is that only the cells on the outside of the aggregate actually end up proliferating. So there's kind of several factors at play, but all of them come down to the competition for nutrients in a spatially structured situation. Okay, so out of that, those simulations, there comes a prediction for the experiments, which is that if you repeat the experiments under different conditions, so you change the density of these single cells, then you should see that when the density is high, the aggregate wins, and when the density is low, it doesn't win. And it turns out you can do that experimentally because when you grow the liquid culture of the bacteria that you seed the flow cell with, you can change the density of the cells, and that amounts to changing the density of the single cells, and so you actually can do that. And our collaborators did, and it turns out the way the simulations predict. So if you do the experiments with a low level of competition, then you see that the single cells actually do better than the aggregate, whereas if there's a high level, then it's the other way around. And so we were kind of happy with that because it suggests that at least basic mechanisms which we have in the simulation explain something about the experiments. So what's also nice about simulations is you can do all kinds of things which are not so easy to do experimentally. And one of the things that Gavin went on to do is to actually say, what if the shape of the aggregate changes? So it's known that that bacteria that adhere to surfaces can produce all kinds of extracellular chemicals which affect their interaction with each other and also with the surface. This is just an example. So this is a picture of a Bacillus subtilis biofilm, which is producing a hydrophobic protein, which makes the surface of the biofilm extremely hydrophobic. And you can see that because there's a dyed water droplet stuck on it, and you can see that it's really hydrophobic. This is actually not affecting the interaction, as far as we know, between the bacteria and the surface, but it's affecting the interaction between the bacteria and the surrounding medium. So it's quite likely that different bacteria might form different aggregate shapes when they stick to a surface, and that's why we decided to explore this, even though we don't have any experimental data on this ourselves. So what Gavin did is he compared an aggregate like this, which is kind of rounded up and forms a rounded shape, with an aggregate like this, which actually spreads out on the surface and forms a much flatter shape, and also shapes in between. And the question is, which of these aggregates is actually better in forming a biofilm, in competing with the surrounding single cells to form a biofilm? And we suppose that how that might happen in reality is that in the rounded aggregate, you might have a repulsive interaction between the bacteria and the surface, which is what makes it rounded. Whereas in the spread case, you might have an attractive interaction between the bacteria and the surface, which is what makes it spread out. And actually, Gavin quantified the shape of these aggregates in his simulation using this angle theta, which is, you can see here. And that's actually, it's called a contact angle, and it's widely used in soft matter physics to describe interactions between soft matter and surfaces. So what he did in his simulations is he initiated different simulations with aggregates of different shapes, so ranging from this very flat one to this very round one. And he also varied the extent of the competition from these surrounding single cells to find out which aggregate shape is best at competing with the single cells. Okay, and so the results of the simulations are shown here. So first we think about the case of low competition. So in this case, there are not very many of these surrounding single cells. And what's plotted here is the success of the aggregate, so the number of progeny from the aggregate per initial cell, as a function of its initial contact angle. So on the left-hand side, the aggregate is initially spread out, and on the right-hand side, it's initially rounded. And what you can see here quite clearly, actually these results are for no competition, but it's also true for very low competition. What you can see here is that it's better to be initially spread out. So the aggregate that's initially spread on the surface does better in the low competition regime. And how can we explain that? Well, actually, it's quite simple to explain, because an aggregate that's spread out, it just has more surface area. And so if you add up the total amount of surface area of this thing that's exposed to nutrient, it's higher if the aggregate's flat. And actually, if you compare the shape of this curve to just a plot of the surface area as a function of theta, it has pretty much the same shape. And so we think that this is just explained by the fact that if you spread out on the surface, then you just have more surface area, which is exposed to nutrients, and you can just absorb more nutrients per bacterium. Okay, but in the high competition regime, so when we have more of these competing single cells, we actually see the opposite result. So in the high competition regime, it turns out that the aggregate does better if it's rounded. So the success is higher for the rounded aggregate. Okay. Why do we think that is? Well, we actually think it's for the same reason as we had before. So the rounded aggregate has a bigger height advantage. So the cells at the top of the rounded aggregate are higher up compared to the surrounding single cells, and so they can compete better. So we think that there's an interplay going on here between the surface area that you have exposed to nutrient, which is bigger if the aggregate's spread, and the height advantage, which is bigger if the aggregate's rounded. Okay. So that brings me to the end of the part one of the talk. So what we found out from these simulations and experiments, we think, is that in biofilm growth, which is a non-equilibrium process, the initial configuration can be really important. And the reason it's important is because you have this coupling between the growth and the nutrients. And actually, this is something that's been known in a different context, not to do with aggregates, but to do with fingering instabilities. And so actually, it's known that if you have a biofilm that's growing and you get a little protrusion, there's an instability which can cause that protrusion to keep on growing. And the reason is that the cells at the top of the protrusion are actually have a greater access to nutrients, and so they keep on growing. And that can happen under some conditions, but not under others. And so we think that what we actually see in these simulations, where we have these initial aggregates, is sort of a variant of this fingering instability mechanism. So if you have a situation where you have an initial aggregate which has a higher access to nutrients, then under some conditions, but not others, it can be that aggregate kind of proliferates and keeps on having an advantage. And we think it's interesting from a physical point of view, but also it could be important biologically, because we know that these aggregates are prevalent in nature and probably quite often are actually seeding biofilm growth. So that's what we learned so far. But we're also interested in biofilms for all kinds of other reasons. And one of the things that we're interested in is how you actually get evolution happening in biofilms, by which I mean evolution of genetic variants within the population. And the reason we're interested in this is because, as I mentioned, biofilms are quite prevalent in an infection situation. So they can form on medical implants, they can also form on teeth, and they can form well, basically on medical implants and teeth. And the reason we're important is because they can be really hard to treat. And so people who have a biofilm infection quite often get many rounds of antibiotics which fail to remove the biofilm. And our question is, what does this mean for the evolution of antibiotic resistance? So we all know that antibiotic-resistant bacteria are a major health problem, but it's not really known what are the factors that actually control the rate at which resistant mutants can emerge in a population. And so, and people who study the evolution of antibiotic resistance, usually in their experiments, they have well-mixed cultures in a test tube. But, for example, Bob here, Bob Austin, has done some important work which shows that if you have spatial gradients of antibiotic, actually that can accelerate the evolution of resistance. And we wonder if in these biofilm populations, where clearly you do have spatial gradients of both nutrients and probably antibiotic, whether that can also accelerate the production of genetic mutants which might be resistant to antibiotic. So we wonder whether these biofilm infections might be a kind of dangerous source of resistant bacteria. So to investigate that in our simulations, we started a very basic question, which is just if you have a growing biofilm, what's the probability that if a mutant arises randomly in the population, it actually ends up taking over the biofilm? Because if you think about the evolution of antibiotic resistance, it's not enough just to get one antibiotic-resistant bacteria in your population. You have to actually have it multiply and take over the population. Because if you just get one resistant bacteria and then it disappears, then that actually isn't a problem. It's only a problem if the resistant mutant fixes, which means it actually takes over the population. And so we've been doing some simulations, some preliminary simulations, where we try to actually calculate the probability that a mutant bacterium which emerges in a biofilm will actually end up taking over the biofilm. So how do we do that? Well, it's quite simple, actually. So we run simulations like the ones I just described, but every now and then we introduce a mutant cell which has a small growth advantage. So as I mentioned, the growth of the bacteria is described by this function, which relates it to the local nutrient concentration. And we just suppose that the resistant mutant just has a larger value of this mu max. So it grows a little bit faster than the surrounding bacteria. Okay, so here's an illustration of that. So here's a biofilm which has mostly wild-type red bacteria, and we introduce a green mutant cell at some position in the biofilm, and then we just run the simulation until one of two things happens. So either the mutant just disappears, so it just got buried deep within the biofilm and it didn't manage to proliferate, or alternatively, it ends up taking over the whole biofilm and the whole biofilm is green, which means that the mutant managed to fix in the population. Okay, so we run the simulations and we do this many, many times so that we can get statistics, and we calculate the fixation probability, which is the number of simulations which ended up green, so the mutant took over, compared to the total number divided by the total number of simulations. Okay, so this is very simple, but there's a practical issue, which is that in these simulations, the number of bacteria is increasing all the time, and so there's a limit to how long you can actually run the simulation for before you run out of memory in the computer. And that's a problem because these simulations can be quite long. And so we have to have a kind of practical solution, which is simply that every now and then in the simulation we just chop off the top and reinitiate. So we say that bacteria which are kind of deep within the biofilm, actually they're not very important because they don't have any chance of proliferating because they're buried anyway. And so, so periodically in our simulations we define the active growing layer, which is the bacteria which are actually growing and reproducing, and then we just do this clipping procedure. So we define the growing layer, we chop it off at its lowest point, and then we simply reinitiate the simulation using only the growing layer. And so, so this is a bit sort of a brute force approach, but the advantage is it makes it feasible to do a really long time simulation of these biofilms, and that's why we do it. Okay, so the, yeah?
Does bacterial death play a role?
Yes. So experimentally, what will happen? Well, when you say, I mean, if you think about a biofilm that's growing on a medical implant, let's say, what happens is that there's a flow field across it, and there's what's called sloughing, which is that some of the bacteria detach from the top. And so the biofilm, at least what we think, is that it constantly gets kind of washed away. And so another way of doing these simulations would be to include that in the simulation, which actually in future we might want to do. In a flow cell experiment, it's less clear what happens because typically the flow rate is pretty low, and so actually they do just get thicker and thicker. So you can argue about whether what we've done here is the most clinically relevant, and yeah, in future we might actually choose to do it differently. But this is what we've done for the moment. So the question that we've been trying to ask is, how does the probability that a mutant fixes in this biofilm, how does it depend on the spatial structure of the biofilm, and in particular the roughness of the biofilm? So if you want an easy way to characterize the spatial structure of a biofilm is by the roughness of its surface. So if you just imagine a biofilm with a surface like this, you can quantify how rough that surface is by taking the standard deviation of its height. And that's what we do. And it's actually kind of a nice thing to do because there's a big literature in statistical physics about growing interfaces and roughness. And people have done things like calculate the critical exponents of how these roughness grows in different models. And so it's nice to make a link to that. And that's one of the reasons why we think about roughness. And so what we've actually done is we've computed this fixation probability for two different parameter sets. For the bacterial growth, which correspond to different values of the roughness, while still having the same active layer width. So we have one parameter set which gives a rough biofilm, and another parameter set which gives a less rough biofilm. And in our preliminary work, what we've done is say, if you have a more or less rough biofilm, then do you get a lower or higher fixation probability? So, and the roughness is defined as this. So as I said, it's just the standard deviation of the height across the biofilm. And the answer is, so far, from our simulations, you actually get a lower fixation probability in a rough biofilm. So it seems that what we discovered so far is that if the biofilm has a rough edge, then it's harder for a mutant bacterium to take over the population than if it has a smooth edge. Of course, we need to do more simulations. We only have a small amount of data and so on, but actually this result, we think it's quite robust. So, okay. So fixation seems to be less likely if the biofilm's more rough. So, yeah?
So, in these rough biofilms, can you also look at the correlation with like the position where you insert this reagent?
Yeah. So yes, that's what we're trying to do now, actually. So what Gavin did in these preliminary simulations is he just picked a horizontal point and stuck it on top of the biofilm. And you can argue all kinds of reasons why that's not a good way to do it, and indeed I did. and what we're trying to do now is a more realistic way, which is that we look at how fast each bacterium is dividing in the biofilm, and we make the probability that it turns into a mutant proportional to that. So we assume that mutants will happen at division events, and so we're trying to do that now. But we don't think it changes this conclusion that fixation is less likely in a rough biofilm. And actually, it turns out this conclusion makes some sense because there's some previous literature which has mainly been done by Oskar Halacek, who's now at the University of California at Berkeley. And what he's been studying is not biofilms, but two-dimensional colonies of bacteria as they grow. So this is a picture from someone else, from Ido Golding's work, actually. So what you do in these experiments is you have a flat surface of usually agar, which contains nutrients, and then you inoculate it with a small droplet of bacteria, and you watch them spread as they grow on the surface, and they spread into a round colony like this. And what this particular picture shows is that sometimes as a colony grows, you get a mutation event, and so you get a sector of bacteria which are of different genetic type from the wild type, and you can see this sector growing here. So these flat colonies are kind of a really nice way to visualize fixation events because you can clearly see the sectors expanding. And so Oskar Halacek in particular has done a lot of work on trying to understand the dynamics of how these sectors grow and when, under what circumstances a sector will actually fix, like you see here, or sometimes you see failure events, so a sector starts to grow and then it eventually gets swamped by the surrounding cells. And what he's discovered from his experiments with colonies is that if the front of the colony, so if the surface of this colony is smoother, then the sectors are more likely to survive. So also in bacterial colonies, fixation events are more likely in smoother growing fronts. And actually, Oskar has a theory for why that is. And what he says is that you can imagine the the growth of these sectors in a one-dimensional model where you have a sort of a diffusion thing going on. So if you think about the edge of a colony, or also about the edge of a biofilm, then you can mark the points where you get boundaries of a sector, and you can imagine these points to be like random walkers on a 1D domain. So you imagine that the 1D domain is the boundary, and these walkers move about. So when two walkers get more close together, that's like a sector is closing. So you have an annihilation event when two walkers collide, and that actually means that you didn't fix. So the mutation was lost because this red sector, for example, disappeared. And so you can write down this diffusion equation with this random walkers, and you can say how many times do they annihilate or not annihilate, and you can use that theory to predict the fixation probability. And what it turns out is that if this diffusion constant along the boundary is larger, then annihilation is more likely and fixation is less likely. And what Oscar says is if you have a rough interface, then the diffusion constant is larger. And actually, he doesn't really know why that is. So the theory is kind of incomplete because the link between the roughness of the interface and the diffusion of these walkers is not completely clear. But nevertheless, there's kind of a link to this literature, which...
How solid is this data, Rosalind? Yeah, I mean, it's a really good solid data.
From Oscar? I think so. I'm not sure. So what Oscar can do really, really well is count the sectors. I'm not sure whether he actually can track so well, for example, the dynamics. So whether he's really able to make a link between this diffusion model and what he sees in the experiments, I'm not so sure.
Like zoom in with the microscope and actually see the actual diffusion of the bacteria and stuff like that?
Well, I think he tries, but in reality, it's not diffusion, of course. I mean, it's a kind of growth.
It's
yeah.
Advective, dynamics.
Yeah. So he tries to do that.
Okay.
Yeah, but I don't think he has a direct link between the diffusion model and the dynamics of the experiment. Okay, so that's what we did so far on this biofilm evolution, and of course there's lots more things that we're trying to do, but we haven't done them so far. So I'm going to move on to a different topic. So the last part of the talk, I just briefly wanted to say some other things. So in the first part of this talk, I talked about aggregates. So you have these aggregates, and they can affect the outcome of the biofilm. But an obvious question is, how do the aggregates form in the first place? And so, and we've been doing some work, preliminary work, to try to understand that. So what we know is that many of these bacteria produce sticky polymers. So, and they're known to be important in biofilm, but they also are important in the formation of these aggregates. And we've been trying to at least start with deciphering the role of what these polymers actually do in the formation of aggregates. And Gavin has actually been doing some experimental work in our lab where what he's shown is that aggregates are actually, polymers are actually crucial in formation of the aggregates. So what you have here is an image from some of Gavin's experiments where he has a Pseudomonas aeruginosa bacteria, and he just grew them up in a liquid culture, and then he imaged them under the microscope, and you can see that there are many aggregates. So here are all the aggregates of the bacteria. And then in contrast, he did the same experiment, but this time with a genetic mutant which doesn't produce polymer. So it's actually got two types of polymer, both of which have been knocked out. And you can see that now there are no aggregates. So what we've discovered so far is that the formation of these polymers is absolutely crucial for the aggregate formation. But Gavin's primarily a simulator, and so the question he asks is, well, how can we simulate that? So in the simulations that we did so far of biofilm formation, there wasn't any polymer production, but now we discover that polymer production, if you want to understand aggregation, you really need it. So, so ideally, what we would do is that we would say, individual bacteria are particles, and they just produce these polymer molecules, and we're going to simulate the polymers explicitly. So something like this. So we do a simulation where we have a bacteria and it produces all of these little polymer molecules, and we can put in some rules in the simulation about how the stickiness works. So how sticky is a polymer when it interacts with the bacteria and so on. And actually we in our previous incarnations, we had done some simulations a bit like that in the previous work, but we only did it for one particle. In this case, it wasn't a bacteria, it was just a colloid. And it turns out that you can't really do this for a realistic number of particles because you just have far too many polymer molecules, so it's computationally unfeasible. And so what Gavin's been trying to do is think of smart ways to overcome this problem. And I just have one slide about how or two slides about how he did that. So what he does is he says, well, here are the bacteria. Here are two bacteria. And each one of them is producing these polymer molecules, which are shown by these little spheres. And let's just try and mimic that by an interaction potential between two bacteria. So we're going to say there's some kind of attractive interaction between the bacteria, and we need to know what the form of that interaction is, because we don't want to simulate all these polymer molecules individually. But it's kind of going to be a kind of complicated interaction, because if you think about it, if a third bacterium comes along here, and it starts to produce its polymer molecules, then the density of polymer molecules in between these two bacteria is going to increase. So this interaction between the bacteria is going to be dependent on the local density of other bacteria, because the more there are, the more of these surrounding bacteria there are, the higher the polymer density we expect to be, and so the stickier the interaction is going to be. And so what this translates into is an interaction potential between bacteria which is dependent on the local density of the bacteria. So actually we've modeled that as a simple attractive Lennard-Jones interaction, but the pre-factor of the interaction is actually proportional to the local density. And in the simulation we measure the local density by just drawing a little sphere around the central bacterium and counting the number of other bacterium that are inside this sphere. So it's pretty simple. And this is really preliminary work, so we don't have a definitive answer to what happens. But what Gavin's been doing is comparing the physics of a system where you have aggregation driven by this density-dependent interaction mimicking polymer-producing bacteria, compared to a situation where you have attractive interactions but it's not density-dependent, which is actually, everybody knows about that, it's a Lennard-Jones system. And what we think happens is that when you have the density-dependent interaction, we think that you get greater control of the cluster size. So as you follow a simulation of these particles in time, what happens is that you get little clusters, which then grow, and you get bigger and bigger clusters, and eventually the whole system phase separates. But if you take a snapshot at a fixed time, then what you see is something like this, so many clusters. And what we think we see in the system with the density-dependent potential is that you basically get clusters which are surrounded by individual bacteria. So you don't see a broad distribution of cluster sizes. You actually see rather uniformly sized clusters surrounded by individual bacteria. Whereas if you don't have a density-dependent interaction, then you do see a broad distribution of cluster sizes. And so our tentative conclusion here is that maybe the density-dependent interaction is a sort of control mechanism, and it allows the system to select the size of the cluster more accurately than if you didn't have this density-dependent interaction. But this is really preliminary work, so hopefully I'll have more to say about that in the future. So that brings me to the end of the talk. So I think what we conclude is that these biofilms are a beautiful example of a multi-scale, non-equilibrium self-assembly system, but they bring with them all kinds of computational challenges. So some of the challenges that I highlighted today are that you have to try and find a way of modeling a growing population in a steady state way so that you can observe mutant fixation. We're thinking about ways to coarse grain these mechanisms like sticky polymer interactions, and of course how to correctly simulate bacterial growth. There are also biological challenges. So one of the things that our work kind of touches on is that these exopolysaccharides, so the polymers that are produced by bacteria, actually they could have many roles. And we're only really thinking about the stickiness, but they also do other things. And of course, there are all these other mechanisms which I described at the beginning and then didn't talk about, like gene regulation and signaling, and all those things we should think about in the future. So here are all the people who actually I didn't mention before but contributed to this work. And thanks for listening.